Hi guys, good evening everyone. Hello. Hello everyone, good evening. I hope I'm audible and visible to all of you. Thank you. Yes, you can write about you. So, okay, we will start in 2 minutes by the time everyone will be joining. There are around 200 participants overall. So we'll wait for 2 minutes. I have sent the reminders to all. So we can just wait for 2 minutes. Good evening. Good evening. Can you please just write about you, what you are working right now, what you are doing right now, in which year if you are studying, and what you are studying right now, and from where you are, guys. If you can just give me a little introduction about you, your name, from where you are, what you are doing, what are you studying, that would be great. Please write about you. So I'll get to know my audience about that. Okay, so please write about you guys, and everyone else will come to know about you in the group, as we all are in the same boat right now. Gaurav Singh, M Farm, myself, College of Pharmacy, Mocha. Perfect. Great, 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 great. Okay. Guys, my team has muted all of you because there was a lot of chaos when we started this. So I hope that's not a problem for all of us. We will definitely unblock or unmute you when the class is over. Okay. Okay. Good evening, Forum Sa. You can write, yeah, Forum. Good evening, Rasid. Okay, Sriya. Are Pritam sir, how are you? Good evening. Abhishek Prajapati, Dr. Mahareen. Fantastic, guys. Nice meeting you. Nice seeing you here in the group. Okay, Akshaya. Dr. Rahila Tarannum. Thank you, Dr. Rahila, for joining. Srishti, Adnan. Okay, Pramada, Dr. Durga Devi. Okay, Dr. Hiriza. Hiriza. Okay. I'm sorry, guys. Sometimes I'm not good in the names. Nupur. Hello. Okay. Some other people were left to join the group. Now they all are in the group, I believe. Okay. Neeraj Chauhan from Biznor, Sita Raman from, okay, Trupur. Okay, so guys, thank you all for joining us in this wonderful journey, the course which we have starting today, pharmaceutical product life cycle, research and development to marketing. So guys, this is a short course, 6 days course, but in the 6 days and almost 8 to 9 hours, we will be learning a lot. Okay, we are going to learn a lot. So we will be, learning about complete pharmaceutical product life cycle, how the product comes, from a very basic chemical entity to a drug, and how it gets into the market and how it reaches to the patient, right? So we will be discussing all of this in these 6 days. In these 6 days we will learn all of these, and I hope you will have a good journey, and you will be seeing today's class. And after today's class, you can invite your friends also if they want to join. Okay, so let's start. Let's not wait much because already there are more than 55% of the participants. So let's join. That's the ideal number to start. Okay, so there are 111 people. So guys, I welcome you again. I'm Dr. Ajit Singh. As you can see on the screen, I'm the founder and CEO of the Curio also, and I'll be taking this entire course. Uh, and I mean, I don't take much classes, uh, for everyone, but this is a topic of interest for me, and I'm into this. So, I love research. Okay, that's why I was very curious to take all these classes, and that's why I came up with this short course where you can just learn. So this is a learning full course, and you will definitely dive, deep into the subject here, and you will be learning all basics, nothing much, all basics. So this is a learning full course. I mean, we are not, you are not going to get any kind of job after this, or we are not, placing you anywhere after this. People usually ask after the courses, sir, are we getting the placement or not? But yes, I'm very sure you will get an immense experience out of it. You will get an immense knowledge out of it, because when I was reading, I read it throughout, and I'm into the clinical research, I'm into the product research, and I know how the practical things work. right? So you are going to learn these things here in this course. So right now I'm working at the Department of Medicine, Kasturba Medical College, Manipal. Manipal Institute, I mean Manipal Academy of Higher Education, previously known as Manipal University, and I'm also working for ICMR as research scientist C. For multiple research projects here, right? And then I'm working for a medical artificial intelligence company as well. So that's our company. So guys, with that little introduction, let me start the things. So before I start, always in my classes, I usually ask some questions to my audience. Okay, before I go further, I have some questions, basic questions, then we can proceed with the class. So there is no conflict of interest, definitely. So in the entire series of 6 days, we are going to learn about drug discovery and preclinical research, and then clinical development of the drug, like drug clinical development, and then all the regulatory bodies and regulatory review and approvals. And we'll be definitely learning about, all Indian regulatory bodies, that is DCGI and CDSCO, and we'll be learning about how it goes to the market and how after the market launch and how they actually plan. So in this, I'm very sure, guys, that all the MFARM, what we say, MFARM streams are included. All the B farms, are included. In the drug discovery, you will be learning the pharmaceutics and pharmacology. Okay, pharmaceutical chemistry also a bit. Okay, and then clinical development is completely pharmacy practice and clinical research, and then regulatory affairs, and then marketing launch. I mean, market launch and commercialization is pharmacy management and MBA in pharma. So these are the all topics what we are covering during this course. So this course is planned for everyone, and everyone will have a comprehensive knowledge of product life cycle, the pharmaceutical product life cycle. Okay, so let's start with a few questions, and I believe all of you will answer. My interns have taken this class already. I have taken these two classes last week for my interns, so I'm very sure they are here to repeat, because today I've added some more interesting facts and some more interesting experiences. They will be definitely taking more in these classes, but they are ready to answer these questions. So my first question to audience is, guys, yes, in which phase of clinical trials is a drug first tested on healthy human volunteers? First phase where the drug is tested in the human beings. Oh. Who is writing phase 4? Wrong. It's phase 1. It's very simple. It's phase 1. Okay. It's phase 1, not phase 4. Perfect. I mean, right answer, because it's a very easy question. Let me take you to the second question. This is phase 1. Answer is phase 1. Now, question number 2 is, what is the primary focus of preclinical studies? What is the primary focus of preclinical testing in the drug development process? Yes, guys. What is the primary focus of preclinical testing in the drug development process? Yes, I'm looking for C, to test the drug in vitro and in animal models for the safety and toxicity. Absolutely. Primary focus of preclinical testing is to test the drug in vitro and in animal models for the safety and toxicity. Because to assess the market point of this comes under the commercialization. I'll tell you all the examples also here. Okay, I'll tell you all the other wrong answers, why those are wrong. Okay. So here, to assess the market potential of the drug means it will come under the marketing and commercialization. Okay, that step comes after the drug is launched to the market or going to launch to the market, right? To evaluate safety and efficacy in humans, that comes under the phases of clinical trials. That is the clinical development phase. Okay, that comes under the clinical development phase. And to determine the cost of production, that also comes under the commercialization before we go to the market, right? And if you want to know about the past things, like phase 1 is the first phase where we just check the drug in somewhere around 1 to 20 or 50 healthy participants, healthy human beings or patients. In healthy human beings or patient, this is the first phase okay, where we expose the human beings to the drug, or we expose the drug to the human beings. That is phase one. Phase two is more like we get more patients, and usually we go to the patients here. Okay, like here we take somewhere around 50 to 300. Okay, and in phase three we take up to 1,000. Okay, I mean phase two, I mean phase four, 1,000 to 10,000. Okay, so this is usually the numbers, and the different designs also. Here we just take either healthy volunteers or patients, maybe in certain cases only patients, and usual cases healthy volunteers. In phase two, less number of patients, or and there is no comparative, usually. In phase three, I mean, there is comparative arms, and usually the placebo. In phase four, the healthy human beings are not taken. Here, all the patients, and comparing the drug, comparing the test drug, or comparing the new investigational drug with what? With the current therapy or already established therapy. There is, either placebo, but here placebo comes down to a very lesser number. Usually here we compare the drug with other pharmaceutical therapies. Okay, so that's how we can differentiate between phase one, phase two, phase three, and phase four. That I'll definitely teach tomorrow. Tomorrow we are, I'll be taking all the clinical phases, but today it's early preclinical and preclinical. Okay, so that's the correct answer, right? To test the drug in vitro and in animal models for the safety and toxicity. Right now, next question for all of you is, which document must a pharmaceutical company submit to regulatory agencies? Okay, guys, listen and focus carefully. Which document must a pharmaceutical company submit to regulatory agencies to request approval for marketing a new drug? Approval for marketing a new drug. Yes. So let me give you two options. First two, either A or B. Yes, guys. NDA, new drug applications. Okay. People are telling NDA, new drug applications. Okay. New drug applications. Why IND? Guys, people are writing INDA. People are writing INDA. No, there is no INDA. No, this is not INDA. Why? Let me explain to you. Let me explain to you, because our learnings start from here only. Okay, see, you have to understand two things here. I'll tell you very important facts, guys. Focus. This is the another class. I will take this slide. Okay, as an another class that will definitely I'll take when I'm teaching you the regulatory part. But today I'll be taking you through a bit, different facts. See, understand one by one. These all four. Investigational new drug. When you're writing investigational new drug, when you're reading investigational new drug, that means the drug has to undergo the clinical trials. Okay, simple. Has to undergo the clinical trials. It has not gone yet. Okay, drug has to undergo the clinical trials first. Okay, all clinical trial phases. Then it will come to the market. New drug application means when the drug has undergone the clinical trials, all clinical trials, and now it is ready to be launched to the market. Okay, so understand, this is preclinical trial phase, this is post-clinical trial application. This is preclinical trial application, this is post-clinical trial application. Understand, guys, this is preclinical trial application, this is post-clinical trial application to the regulatory authority. Okay, so it's very simple to understand. When we talk about investigational, that means the drug has not undergone any kind of clinical trials. So first it will go through the clinical trials, and then we will launch into the market if everything goes well, and then we will be filling the NDA. So approval for the marketing comes under the NDA, not IND. Okay, and pharmacovigilance report even comes after the launch. Okay, this is post, this is post marketing. Pharmacovigilance report comes after post marketing. I mean, after the marketing only, after the drug is in the market and people are taking, and then they are, reporting the bulk side effects, or any kind of side effects. So that is the observation once the 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 drug goes to the market and everyone is taking. Everyone means every patient of the relevant condition, they take it, and they collect the mass data from the public. Okay, that is pharmaceutical report. And now clinical trial protocol. Okay, clinical trial protocol is the document. Okay, that is not the application, that is the protocol. That is the protocol or the document or the plan to conduct the clinical trial, that will also be submitted to the regulatory agencies, but before clinical trial, not after clinical trial. So protocol will be usually submitted to the IEC and regulatory authorities, right? Before clinical trial, not after clinical trial. Understand? So I hope it is clear to all of you. Basic. Is it okay? So the correct answer here is B, not A. Okay. The correct answer here is B, right? B is the correct answer. New drug applications. Okay. New drug applications. Now, next, I believe, I hope we have done with the questions. No, I think there is one more question or two more. Okay, but that's okay. I mean, these are the good learnings. What is the main purpose of phase 4 in drug life cycle? What is the main purpose of clinical trial phase 4, CT phase 4 in drug life cycle? To test the drug's efficacy in a small group of patients, to conduct additional studies for long-term safety and effectiveness after the drug is marketed, to determine the initial dosing requirement, to evaluate the pharmacokinetics of the drug. Definitely it is B. Definitely it is B, because I just told in the last slide. Okay. Will not waste much time here. Okay. Okay. Now I think that's done. That's the last thing. Which stage involves scaling up manufacturing and establishing the distribution channels? Which stage involves scaling up manufacturing and establishing the distribution channels? Yes, guys. Yes, yes, first. C, market launch and commercialization. That also we have just discussed. Okay, that also we have just discussed. Okay, so now we are here with the class. Now we are here with the slides. Okay, now we are here with the slides. So let's start with that. Today, we will start with the today's class, drug discovery and preclinical research. Now, guys, I want your 100% attendance, attention, and attendance also, 100% attendance and attention, and you have to be interactive. Okay, not in the mic. Definitely, we are not going to open the mics right now, but you have to be 100% attentive with your fingers and writing the text. Okay, so let's start. So there are four things. Okay, in today's class, we'll be discussing about four things. First is target identification and validation, then hit identification and lead optimization, then preclinical testing and safety and toxicology studies. Okay, this is called drug discovery and preclinical research. And in that also, there is something called early preclinical research. Early preclinical research, I'll come from the next slide. So here, this is the summary of the entire class, what we are, going to learn today. Okay. So can someone tell me these three terms? There are three terms what I am not understanding. One is target, one is hit, one is lead. One is target, one is hit, one is lead. Okay. So can someone tell me without looking at the screen, like what is target exactly? I mean, what do you mean in the simple words? Here I can say identifying a biological target, whether this is protein associated with disease, validating it with the potential therapeutic intervention. I didn't understand it. Can you tell me what is target? I will explain everything very nicely to you. Don't worry. What is target, guys? Target? Anyone? Receptor? Okay, the disease or microorganism responsible for it? No. Receptor? Okay. See, guys, you can write your things. Let me tell you. I'll give you an example here. I'll give you an example here. Okay. So suppose someone got diabetes. Diabetes is a very new disease. Just you have to imagine. Okay. Diabetes is not new, but diabetes is very common. So everyone knows about diabetes, right? Everyone knows about diabetes. So let's understand, diabetes is just newly detected. There is one person, guys, you will understand everything here with this example only. There is one person, you know, detected with, suppose, high blood sugars. We know blood sugars, but there is one person who is detected with the RBS of 380 milligram per DL. Okay, his blood sugars are very high, and this is something crazy. This is something crazy. Why it is happening? We have no idea why it is happening, right? We have no idea why this blood sugar is so high. Correct. We don't know that why this blood sugar is so high. Okay. So what scientists will do is they will understand the cause, the root cause analysis they will do. Okay. They will do the root cause analysis, why it is happening. So what they do exactly, guys, focus here. What they do is they will find out, they will compare the person with a normal person, with a healthy person. Just compare, like, what have changed in this person. I'll call it patient. Okay, I'll call him patient right now. Okay, and there is a healthy person. Guys, this is the basics. You will understand everything. There's a patient and there is a healthy person. So they will see what happened in this person. So his RBS is suit up to this level, and what's not there in the healthy person right now. What is different in the patient? Okay, so they will get to do all the tests, all the relevant tests, and they will see for proteins. Okay, they will see for genetic changes. They will see for receptors. They will see for all, the tissue biomarkers, right? And they will see, like, in the proteins, what they will see: overexpression or underexpression of any protein. Usually overexpression or underexpression of any protein. They will see how the proteins have overexpressed or underexpressed in this condition. Okay, why this is overexpressed or why this is underexpressed. They will just look at that. Okay, and they will look into the genetic modifications, any type of genetic overexpression or underexpression or genetic modifications in the patient. They will look for all the other parts like enzymes. Okay, if there is any enzyme raised or decreased, right? There are multiple things. I'll tell you everything. Okay, there are multiple things. So they will find out something, that okay, enzyme. Okay, there is enzyme, or there is a receptor which is overexpressed, or the protein is overexpressed, right? They'll go, they will find out, okay, there are some problems in the pancreas. Okay, pancreatic cells are dying. Okay, pancreatic cells are damaged, or pancreatic cells are not producing the insulin. Why they are not producing the insulin? Beta cells, right? Beta cells, why they are not producing the insulin? They will see what protein, what genetic receptor, or I mean what genetic modifications has been done, or what kind of receptor or enzyme is involved. Usually there are four things: protein, genetic, receptor, or enzyme. Usually, I mean, receptors are also protein or enzyme or tissue factors. Okay, so they will look for that. They will look for that, what is overexpressed or underexpressed, what is underdetermined or what is over determined, right? So now they will see that, okay, in this, something has raised up. Suppose I'll give you, there are multiple drugs now. There are multiple drugs. Why I've taken the diabetes as an example? Because there are multiple examples. DPP4 inhibitors, glitazones. Okay. There is insulin, right? There is metformin, bigonides. There is, what we say, SGLT2 inhibitors, right? Multiple things. So suppose there is a sodium glucose, transporter inhibitor, transporter receptor is overexpressed. SGLT is over. This is a very new drug. Okay, though I mean SGLT2 is overexpressed or SGLT2 is overworking, right? So that's why the sugar levels are going high, and that is also relevant or related to the insulin resistance in the body. insulin resistance in the body, right? Or similarly, when we talk about insulin, what insulin does is, what insulin does, insulins usually reduce, cut down your blood sugars, right? And that will help. This is a artificial insulin which will help you maintaining your blood sugars. But there are the receptors, there are the degradations of the insulin in the body, and then the degradation of the sugars in the body. So there is a mechanism of actions. I will not go to that, but I'll just tell you how they find it, right? They found that SGLT2 is overexpressed here, or it is overworking here, right? So now SGLT2 is their target. You understand? The target is whatever has been changed in this condition, whether that is protein, gene, enzyme, tissue, or whatever. Okay, whatever is changed or whatever is new happening in the body, that is our target. Okay, that is our target, right? So any kind of biological target usually the proteins associated with the disease, those could be targeted now with the new thing we call drug, right? That thing we have to correct. So the biological targets or biological proteins, genes, or anything which can be corrected to correct the disease is target, is target. Understand? Clear? Target. I hope target is clear now. Now let me come to the second part, hit. Okay. Now what happens is you got that in the body, SGL, terminal part is overexpressed or overworking, right? I'm just giving an example. Okay. Now what you will do, like, array, let's see what exactly is SGLT2 and how many amino acids are there, how many, then you will go to the chemical structure of SGLT2. Okay. What type of protein or whatever. If the gene is there, then you will look into the, DNAs, you will look into the mRNAs, you will check whatever is there inside that, and then you will target that. So once you got a target, you will go back and you will find a chemical entity. There are thousands, millions of chemical entities. There are millions, not millions, billions of the chemical entities. Means, I mean, there are certain, we have in our periodic table certain number of, metals and certain number of chemicals. But with the combinations, with the water attachment, with, I mean, a lot of things are there, right? I am not going to the chemistry. So you can get any structure, billions of structural combinations you will get. Okay, so now you will go back and in the databases, with the help of computers, with the help of analytical tools, with the help of laboratories, you will find that which is the most suitable chemical entity which can help us getting down SGLT2. Understand? And that chemical entity will be called HIT. That chemical entity will be called HIT, and this HIT is going to become the drug at the end. Okay, when it is going to the market, this HIT is going to become the drug. But not only this, in the first, when we are just identifying the HIT which is most suitable to SGLT2, the number is not only one, number is 10,000 almost. On an average, it is 10,000. We find out 10,000 hits, almost 10,000 hits just to tackle one situation. Okay, just to tackle one situation or multiple things related to that, we get 10,000 hits. Okay, and then this hit become, after refining, after getting the most attached hit to the target, we usually get the lead. The hit becomes the lead later. Okay, hit becomes the lead later after refining. Okay, after refining their potency, efficacy, safety, and everything, the chemical entity first is hit when we are just matching, and it will become the lead in the later phase. And that will become the drug after clinical trials. Okay, so it will start with the 10,000 of entities. It will come to the 100 of leads, and then, sorry, 100 of leads, and then it will come to the one drug at the end. One drug at the end, or maybe 3 4, like in SGLT2 there are 3 4 chemicals. One is empagliflozin, and there are others, right? Gliflozins, there are multiple gliflozins, right? So you can get many. You can get many of these, but not exactly 10 or 15, but 1, 2, 3 drugs at the end, and those all should go through the clinical trials, and then only they will become the drug. Understand? So hit will become lead, and lead will become drug at the end. And first thing, you have to find the target, then the hit, then the it will become lead, and then everything will be optimized in the clinical scenarios. Not clinical scenarios right now, we are talking about preclinical scenarios. So once you have the hit, once you have the target, once you have, I mean, the lead, then what you will do is you will refine more. You will try to get the best combination of lead and target. You will try to get a best combination of lead and target, and then you will refine it more for the chemical, what we say, that chemical properties, stability and all. You will look into the other properties, physical properties of the entity. You will look for the chemical properties of the entity. You will look for the efficacy, toxicity. You will improve that hit to become a lead, and you will make it most suitable to go along, get along with the target. Understand? That's how it works. That's how it works. And we are going to discuss this only in the upcoming slides more deeper, right? And once everything is done, you have the target and you have the lead, then you will go for the preclinical studies, right? Now the time is to come out of the laboratories. Laboratory means the chemical analytical laboratories, the chemistry laboratories and analytical laboratories. Right now, till now, we were in the chemical, I mean, the chemistry or analytical laboratories. But now this will be the time to go to the pharmacology laboratories, or to the animals, or like in vitro or in vivo conditions, right? So we will have the targets in vivo or in vitro, the cell cultures or the animal models, and we will try the lead in the cell cultures or animal models. Now in the preclinical testing, we will be checking what? We will be checking, the ADME, the pharmacokinetics and pharmacology, pharmacokinetics and pharmacodynamics of the drug, and of the cell culture or whatever we are using, just to check if this drug, if this lead is suitable to check or suitable to go into the human beings. Understand? So that how it happens is, it takes time, and now it is preclinical studies. Preclinical means before going to the human beings, we have to check in the similar content which human beings carry. Right. We have to check this in the similar content what human beings carry, the cells, the models, the system, and the tissues, or whatever the parts of the body. I mean, wherever it is going to work, targets, right? And then once those things are there, we will look for other studies as well, the safety and toxicology studies that we are going to discuss. So this is the whole thing here. First thing is biomolecular target. We get a target, right? Whether it is from the gene, whether it is protein, whether it is any kind of biological information, or whether it is a receptor or pathway. Bioinformatics and diseases, like whether it is a pathway which is related to the disease. So we target the pathways also, pathways or proteins or genomics, or the cell targets, right? So we can target these things. And then we go to the HTS. This term is called HTS, high throughput screening. High throughput screening means here we optimize our search, or we search for the hit. We search for the hit, and hit we can search through screening, the optimization, combinational chemistry in the labs, and setting selection parameters, computational screenings, chemical screenings. So we check for most combined or most aligned a chemical entity which can go along with targets, and that we will go to the leads from there, right? So through HTS we get our hit here. Then we go to the lead by synthesis, isolation, structural simplifications, structural activity studies like stability and all, physical stability, lead optimization, library development for the same lead, because we don't have only one drug. Right now in the lead time, we have at least 1,000 to 5,000 of the drugs, 5,000 of the leads right now. Okay, then it will go to the preclinical study where we check in vitro or in vivo, cellular disease model, drug selectivity, animal models, and then clinical trials in the human beings, then regulatory affairs, and then we get the approval to go to the market, right? So this is the overall picture of a clinical development. Okay. Now, today we are going to discuss early drug discovery. Today, we are going to discuss the early drug discovery. So first thing, target identification and validation, and then hit identification is the second step. Hit identification. There we go through the high throughput studies, right? And then there are multiple ways, virtual screening or AI/ML, protein expressions, assay development, compound sourcing. I mean, there are many things, go through the catalogs and all, right? And when we go from the hit to lead, mean from the 10,000 hits to 1,000 leads, usually we go by the different catalogs, most suitable library design, and most suitable custom spaces for certain leads and targets. And then computational chemistry support also we take. And then once it is there, we optimize the lead for the suitability or druggability. Now at this stage, the stage is called actually the druggability testing. Sorry, druggability testing. Okay, to check the druggability of the lead. Druggability means how it can become a drug. So to make this as a drug, to take it out, this lead as a drug, we have to check for appropriate target or pathways. We have to check for potency, which is most suitable lead, and we have to minimize the toxicity. We have to go through the preclinical candidates. We have to go for toxicology studies, multiple type of things we have to do, and at this stage. And then we go to the preclinical studies where we have, this is also called as pre-formulation. Here we synthesize the drug. Here we check for the toxicology. Again, we minimize the toxicology, the toxic components or toxic properties of the drug or the lead. And then we validate the method so we can start manufacturing, at least for the clinical trials. And we check for the stability and degradation studies also in the preclinical phase, right? So let's start with first step. Guys. Did you get the summary? Did you understand the summary till now, whatever I have gone through? If you have any doubts, please ask me, because now we are diving more with the examples. Yes, guys, any questions till now? Anything you want to know more from my side? Or it's clear? Yeah, hormones too. Yes, absolutely. Is there anything, guys, you want to ask, or all clear? If clear, just write, and so we can go ahead. Is this topic interesting, guys? Just asking you. I mean, definitely, if it is interesting, then only there are people. There are almost 200 people. So, is this interesting? Okay, great. And it's on me also. I have to make it more interesting. That's okay. Okay, fine. So you are actually, developing a drug here. You're actually developing a drug here in this. Okay, combining all physical pharmacy, pharmacology, pharmaceutical analysis, and chemistry. I mean, no, we are not going very deep, but we are finally making the drug here. We are actually coming up with a drug and steps of the drug, right? So the first step we start with is target identification, and we have to validate the targets. So see, target identification and validation are early and essential stages in the drug discovery, definitely. And they usually, as I told you, they usually have proteins, gene, enzymes which are playing the crucial role in the disease, and then confirming the modulating you know the therapeutic effect. So if we are targeting these things, okay, let me go further. I think here you will see target identification strategies. What are the suitable targets, or what are the usually, we focus when we are finding the target. We have to focus on multiple things. So first thing here is, if you can see, there are few things. One is, let's start with here, pathway. Okay, pathway analysis, right? Pathway analysis. So what exactly is pathway analysis? Target we are searching for, pathway analysis. Can someone tell me a bit about pathway analysis? What do you understand by this term pathway analysis? I'll give you a perfect example so you can understand it best. Yes, guys, pathway analysis. Who will tell me about pathway analysis? Will not get a chocolate for sure. Yes, pathway analysis is what, according to you guys? We have a lot of things, gene expressions, we have proteomics, we have functional screenings like overexpression of the genes, overexpression of the RNA, genetic association, system biology. We have a lot of things here. But I want to know pathway analysis, the metabolic pathways through which a drug shows its therapeutic effect. Perfectly fine. Absolutely understanding. Okay, understanding how the disease work or mechanism of the disease. Absolutely. Here we will look for the disease root cause. See, now I'll give you an example. You all know hypertension? Absolutely, you know hypertension, right? So in the hypertension, there is something called RAS pathway. We also call it RAS system, isn't it? RAS system. You know RAS system, guys? I'll explain it to the words like here. I'll just write the full form. That is renin, angiotensin, aldosterone system or pathway, right? Renin, angiotensin, aldosterone system or pathway. Specific pathway in mechanism of action. Okay. Renin, angiotensin, aldosterone system. Correct. Now, let me tell you, you will understand how the target is identified as a pathway here. See, so if you see that how exactly it starts. Okay, there is a kidney, right? Kidney, when your blood pressure is high. We are talking about hypertension, right? I'll not go deep into the hypertension because all of you may not understand that. See, when there is hypertension, overall, I'll tell you, your peripheral resistance will be high, right? Peripheral resistance will be high. And that will be peripheral resistance means output and, there are multiple other things what you will, multiply and then it will become the peripheral resistance. Peripheral resistance will be high. Hypertension means your peripheral resistance is high. When your peripheral resistance is high, then normal, you have the high blood pressure, right? So in that case, what happens is in the kidney from its glomerular, layer, it starts releasing the renin. Okay, it starts releasing the renin. It starts releasing the renin, and this renin actually will go through the main circulation. It will go to the lungs. Okay, it will go to the lungs, and there in the lungs, it will, activate in the circulation also and lungs also. It will activate actually, yeah, in the circulation only, it activates what? Angiotensin enzyme, angiotensin converting enzyme. We call it angiotensin converting enzyme. It activates the angiotensin converting enzyme out of the lungs and in the lungs. So when it reaches the lungs, angiotensin converting enzyme in the lungs. Now, this was the kidney in the circulation and lungs. Now in the lungs, what happens is there is angiotensin 1. Okay. And then this angiotensin converting enzyme will convert angiotensin 1 into angiotensin 2. I mean, deactivated form to, I mean, the, dormant phase to the activated phase. Okay. Angiotensin 2, and that will help aldosterone, more aldosterone to be released through the sympathetic nervous system, through the sympathetic nervous system. Okay. And once it is there, again, it will help in increasing the sodium and water retention in the circulation. And again, it will raise the BP. Understand? That is the usual pathway. That is the usual pathway. Okay. But now, how, I mean, this is happening. The scientists read it, scientists worked on it, and they found that this is the pathway. We have to work something on the pathway. So what they targeted is that this is the pathway, and in this there is a specific thing called angiotensin converting enzyme, right? Angiotensin converting enzyme. And then they found the hit to tackle the angiotensin converting enzyme, and they came up with ACE inhibitors, ramipril, enalapril, fosinopril, lisinopril, all the drugs. Ramipril, the first drug usually. They came up with the pril family, just to tackle the angiotensin converting enzyme. Now they can stop yeah, sorry. Yeah, now they can, yes, correct. I'm right. So they will just stop this angiotensin converting enzyme. So angiotensin, which is in the latent phase or which is not active, will be similar and it will not activate to angiotensin 2. So angiotensin 2 will not go to the aldosterone. Not going to the, not increasing the release of aldosterone and not increasing the blood pressure ultimately. Understand that. So this is how they target the pathway. This is how they target the enzyme. This is how they find the target. This is how they find the target. So target could be anything. Target could be anything. It could be a pathway. It could be the proteins. It could be the enzymes, right? It could be the enzymes. Or if there are some genetic problems, if there are some genetic problems, genetic association, genetic overexpressions, mRNA overexpressions, so they try to come up with certain drugs based on the target. Like in cancers usually proteins and genes are overexpressed, so they come up to suppress those genes with the drugs, right? So there are multiple targets. You can see here multiple targets and multiple strategies. You can check one by one when when the scientists target that. You can go through the literature, you can check the system biology for brain natriuretics, you can check for the genetic associations, you can check for the overexpressions of mRNAs or functional screenings, you can check for the pathways, you can check for the proteomics, you can check for other genetic expressions. There are the few things what I can list here. These cover everything usually. Okay, these cover everything: protein, enzymes, genes, sorry, G is genes, and then pathways. These four covers all type of targets in the body. Okay, so this is how you find the target. Okay, this is how you identify and find the target. So once you get the target, what are the steps actually? So this step involves selecting a specific biological target that is believed to be involved in development or progression of a disease. Understand what I told you? This is the target. This is the target what comes under the system biology. System biology is enzymes and all. System biology is enzymes and all, and the specific tissues. Like if I talk about insulin, see, insulin is not directly working on any enzyme or this. Insulin is working on the biological system. It is replacing the natural insulin which was supposed to produced by the beta cells in pancreas, right? So that is system biology. That is system biology. Okay, that's the example of a system biological interference, right? So we are replacing something directly. We are replacing something directly, and the insulin synthesis was done from someone else, not from the human beings, right? It was we got it from the cow, from the pigs, right? So we got that and we replaced the human insulins just to manage the sugar levels in the body, right? So that's the system biology. Okay, that's the system biology. But other than system biology, there are many other things. These are the very rare examples of like system biology changing and all are very rare examples and are breakthrough in the science and medical science, right? But usual things, what we usually target are genes or proteins or enzymes or the pathways at the end, right? So this is like the target. We found the target. That could be protein, gene, RNA molecule, or even the biological pathways, right? So the main goal is to find targets those are druggable, where we can actually work to tackle the disease or to interfere with the disease process, where we can actually stop the disease processing here. Understand, we can stop the disease processing here by the lead or hit anything, but first we have to find the actual target, we have to find the most suitable target where we can hit and we can suppress the disease by targeting that. Understand? So now target identification validations, there are multiple validations. Means we do multiple studies and we find out which is the most relevant target, okay. Here we check for the most relevant target. Most relevant target means, see, never ever, there is never ever a single cause for any disease. Never ever. There is not a single change in the body when you get a disease. There are thousands of the problems. There are thousands of the target. Thousands of the target. Okay. There are thousands of the target. Suppose I'll give you an example here. You'll understand more. What is the why we are focusing more on the target identification and validation? Why we are focusing on that? See, I was giving you the example of diabetes only. Okay. So here there is biguanides, there are insulins, there are DPP-4 inhibitors, there is SGLT2 inhibitors, there are glitazones, multiple drugs are there. Multiple drugs are there, right? So means there are multiple targets, there are multiple reasons, there are multiple overexpressions, underexpression, changes, everything. So we have to target one by one. Most importantly, first target should be which is most relevant, so understand here. See, I'll give you the most suitable example also here. See, you people know about MI, myocardial infarction, right? Infarction, or we call it heart attack, right? Tell me, what are the two most important drugs to tackle the heart attack, or we give in the heart attack patient? Two most important drugs. Yes, guys, you only tell me. What are the two most important drugs we give in the heart attack patients? Yes. Yes, guys, tell me. Yes, two most important drugs in the heart attack. Aspirin, statins. Statins are not for the heart attack, dear. Statins are for the hyperlipidemia, which is underlying cause for heart attacks. I mean, just correcting you, nothing else, okay? Yes. Nitroglycerins are symptomatic relief. Okay, I got two wonderful names here. Nitroglycerins, isosorbide dinitrate are not the drugs for heart attack relief. I mean, those are symptomatic relief. Okay, we just relieve the angina with nitroglycerins or glycerins or nitrates. Okay, nitrates are not the solutions. Nitrates are the symptomatic solutions or symptomatic reliefs. Nitrates are the symptomatic reliefs because those are vasodilators. Understand? Just correcting you all, guys. Isosorbide nitrate or all the nitrates are just the solution for symptoms because these are vasodilators. These actually increase the parameter of capillaries inside, so it can reduce the blood flow, it can reduce the blood pressure, and that can help you reducing the angina. I mean, it can increase the oxygen supply to the cardiac muscles, so you can get relief from the chest pain. That's actually the nitrates work. How? Okay, that's how the nitrates work. Metoprolol is again the vasodilator, beta blockers. Digoxin, no, digoxin is a heart failure drug, guys. Streptokinase is emergency medicine, emergency thing that is called thrombolytics. Okay, yes. So someone has written two names. Let me come to that. Otherwise, it will go long. See, aspirin, clopidogrel, right? Aspirin, clopidogrel. The most important thing in the heart attack, guys, let me tell you, I mean, it takes a lot of time when I teach. You have to be with me, patient. Okay. So, see, when there is heart attack, what exactly happens is platelets are most affected particles in the body, most affected element in the body when you get the heart attack. And platelets are the most important reason for heart attack. I mean, mechanism of action when I'm talking about, not the reasons, not the etiology. I'm talking about mechanism of action. So platelets actually, gets aggregated. Aggregated means all platelets, almost most of the platelets come together and make a plaque, like with what we call it clot, right? What we call it clot. Platelet aggregates with the help of what is that? Von Willebrand factor or collagen. Okay, von Willebrand factor and collagen, and what else? What else? Fibrin. Okay, fibrin. That's all, right? And there are multiple other things, collagen and GP2B. Okay, GP2B, like glycoproteins, GP, glycoproteins. These are the receptors, glycoproteins, von Willebrand factors, which are actually located on platelet surface. Okay, these are located on the platelet surface. And when there is something happened inside your cardiac vessel means the vessels inside the heart, which actually carry the blood to the heart and heart muscles, right? So there is something injury, like suppose let me give you the very basic example. Suppose there is a pipe, and in that pipe water is flowing, right? And then some other organ, I mean some other components, toxic components, and something which can what we say, which can destroy the pipe, right? So inside that, which can destroy the capillary wall, or which can injure the capillary wall, like the toxic components or catheters or calcium or anything, or the fat particles, those come and those injure the capillaries. And from that injured from that injured area, these all come out. These all come out: glycoproteins, von Willebrand factor, collagen, fibrin, and all. I mean, it's a big process again, but these all come out, and these all catch the platelets which are going through the circulation system. These catch all the platelets and make a plaque, make a clot. These all make a clot. Understand? These all make a clot. Now, why I'm telling you this whole background story? Why I'm telling you the whole mechanism of action of target, I mean, clopidogrel and all, how the heart attacks are happening. Now the thing is, let me come back. Aspirin and clopidogrel. Okay, clopidogrel. See, suitable target identification we were talking about, most relevant target identification we are talking about, right? So how it happens? When we checked why the clot is happening, why the clot is being created, so we found that COX, coccinase, okay, COX is the enzyme which actually helps all the receptors to bind with the platelets, because platelets also have these receptors in the, I mean, on their surface. COX. Then P2Y12 is another enzyme. P2Y12 is another enzyme, right? I'm telling you, these two are most, there are several enzymes and list of coagulation factors. Lot of things are happening, but almost 60 to 70% of the clot is becoming because of COX. Then 15 to 20% or 15 to 30% of the clot is happening because of P2Y12 enzyme. Okay, because these two enzymes are most relevant and most interfering enzymes in clot preparation or clot creation, or in the, I mean, in, if you go to the mechanism of action, they are helping all the platelets to aggregate. These are helping all the platelets to aggregate. So here we came up with COX inhibitor, aspirin. Aspirin is COX inhibitor, right? Aspirin is COX inhibitor, right? So aspirin is the only drug in this category till date. You understand that? Till date, only the hit which become the lead and which became the drug now is aspirin. Okay, none other than aspirin is there in this category till date. None other than aspirin. So aspirin is the most suitable hit, and COX is the most suitable. COX is the most suitable target here. Understand? So it will cover 60 to 70% of the things. Sorry, I'm so sorry. Right? And then now, if you look at that, see, 30%, 15 to 20% of the work or 30% of the work is not less. Then they came up, they were like, let's close almost all the pathways. Let's close almost all the pathways. So they came up with clopidogrel, which is P2, sorry, ayo, P2Y12 inhibitor, right? Which is P2Y12 inhibitor. So this is the second most relevant target here. That's how we come up with the targets, guys. That's how we find the most suitable target and we validate. Validation means I'll tell you how what gets validated. Okay, so that's how you find a target and validate it. This is the most suitable targets. Okay, and then we modify a lot. I'll come to that later. Okay, so here I mean, to get the target, we do the genomic studies to find out the gene. And example here is, I'll not spend much time now. Example here is mutation in the BRAF gene in certain cancers, mostly in breast cancers. BRAF gene. Proteomics, studying proteins to find one that are upregulated or downregulated or mutated in the disease. We check proteins for upregulation, downregulation, or mutation. Okay, we check the disease for, I mean, we check the genes for mutation. We check the pathway, biological pathways which are involved in the disease progression, right? To the, and then like examples here is, in cancer research, scientists identified HER, which is a growth promoting protein, as a target in certain types of breast cancer where it is overexpressed. HER2 is overexpressed and that contributes to uncontrolled cell growth. That contributes to uncontrolled cell growth means the cancer. Okay, so HER2 is our target. HER2 is our target. And abciximab, if you have heard of abciximab, right? Abciximab is the drug which is the hit or which is the drug for HER2. Okay, which is the drug for HER2. Now, target identification is done, and common methods for assessing the druggability of a target is sequence based. Again, the predictions are solely based on sequence features of the target. Examples like we can have the targets, LNCT, RNA targets and all. Structural base, like we have the 3D structures of the target. We create the 3D structures of the target. See, this is the assessing the druggability of a target, like how we can target the target to make a drug, to come up with a drug, right? So structural base means we create the 3D structures of the target and we try to fit that with multiple chemical entities which fits more there. That will be our hit. Okay, then ligation based. So there are the ligations and we can have the drug ligations like Target Hunter, PubChem, and Binding DB. These are the softwares which we use usually. And then the predecessor based, there are multiple things to assess the druggability. Now the second step is hit identification, right? So hit, what is it? I told you the early stages of drug discovery. It involves the finding initial compound or initial chemical entity, which is known as hit, that shows activity against a specific biological target. So hit is something, the chemical entity which shows the activity against a specific biological target, such as protein associated with a disease. I mean, targets, then defining a target. If you define the target, scientists first identify a biological target, as we told, like an enzyme or receptor that plays a critical role in disease. For like example, enzyme BCR-ABL in chronic myeloid leukemia is target because inhibiting it could prevent the cancer cell growth. Okay, so target, first we define the target and we find the hit. Then now we go for the screening for compounds. Hit identification after defining the target, we go to the compound screening or finding the hit. So there is a library of chemical compounds where billions of compounds are there, billions of compound combination, not only 10,000 or 100 or saying. See, there are limited number of compound, 30, 40, 50, right? But there are the combinations, those make billions of the combinations after modifications, instances. These all makes billions of the combinations. Okay, those are screened and to find the molecule which interacts with target in a desired way. interacted with the target in a desired way means which is most suitable hit. So we when we are finding the hits, there are almost 10,000 hits we take, which can inhibit the enzyme or blocking the receptor or whatever is most relevant, right? So there are two mechanisms usually. One is high throughput screening, one is virtual screening. So high throughput screening is common technique which thousands to millions, guys, look at this, thousands to millions of compounds are tested quickly to identify those which potential activity against the target. Those have the potential activity. So high throughput screening means again we are using all the computers, all the high end computers in the laboratories, in the in our labs, pharmaceutical labs, and in our pharmacology labs, and pharmaceutics labs and all. We do that, and you can tell me, there are what we say, in the drug discovery there are multiple softwares we use in our pharmaceutical colleges also, right? There are multiple softwares, right, for the drug discovery we use. So those can be used for the high throughput screening, where we just put our target and we find the most suitable hit. Okay, most suitable hit against the target. There are virtual screenings also. Again, we are using the computational methods only to screen compounds in silico method. Okay, in silico method, allowing the scientists to predict, a compound. If the compound is not available, see high throughput screening means we have the library, millions of the compound, and out of that we find the hit. But virtual screening means we don't have the hit with us. We don't have the hit with us. In the virtual screening, what we do is we predict. And there are the softwares for drug discovery which helps us, like in finding the virtual hit, and then we create the compound or we create the chemical entity accordingly. Once we get a hit target hit which is virtual, which is imaginary actually, which is created by the computer, then we try to make a compound like that, looks like that, having the property like that, and that also can be converted into the leads later. So hit identification can be done through two methods. One is high throughput screening, STS, another is virtual screening. Okay, guys, understand? Hit identification and identifying hits are again seeing identifying compounds that show the desired biological activity during screening are identified as hits. Hits typically demonstrate the measurable binding or inhibition the target side. So desired means we need something which can be measurably binding or inhibiting the target site, right? The compound must meet the minimal criteria for activity, potency, and selectivity. There is a eligibility criteria for the identification. Why? Out of millions or billions, we just take thousands or 10,000s. What 10,000s? Which are meeting the criteria. We keep the criterias of like they are active against the target, right? Either inhibiting or enhancing or whatever they are doing, enhancing the pathway, restricting or whatever they are doing, they're active in form. Okay, they are potent, they have the power to do the job, whatever job will be defined, either inhibition or suppression or whatever, or whatever type of things we are expecting from that, it should be potent. And then it should be selective means, see, there is one thing very important, you have to understand the selectivity means, see, if we are giving one drug, it should hit only that target where we are actually looking for. It should not affect, if we are giving the drug for the receptors in the pancreas, it should not hit the liver, it should not hit the, I mean, toxicity is there everywhere. I mean, because the liver works in the absorption, and kidney works in the elimination. So toxicity can be there in entire body, but the selective action must be there only with the similar proteins as we have the target, or as we have in the target, right? So this compound, the hit should meet the criteria for activity, potency, and selectivity, right? Hit may have a wide range of potency and effectiveness, as this is just the initial phase. of finding a promising compound. So that's what I'm telling you. There are almost 10,000 compounds which are eligible to become the hit, right now. And then we are confirming them and validating. How we are validating? Hits are retested again and again to confirm their activity and to ensure that the results were not due to experimental errors. This might involve repeating experiments or additional methods to verify the results. So we verify and we validate the hit to become a lead multiple times, 1,000 times in the laboratories, 1,000 times. Okay, not only 1, 2, 10, 15, maybe 1,000 times. We we retest the hits, and we do it on the targets, and we try to get the most suitable results here. Like, it should not be an experimental error. It should not be because of the other chemicals. It should not be because of the machinery. It should not be because of the instruments. It should not be because of the environmental factors. It should not be because of microbiological factors or anything. It should be just because of hit. The target should respond to the hit only, not the other factors. And once you are proving that, that is called hit validation. Okay, that is called hit validation. Okay, so example of hit identification. So in case of HIV drug discovery, researchers targeted the HIV protease enzyme, essential for viral replication. By screening the compound libraries, scientists identified initial hits that could inhibit the HIV protease. This hit were then optimized into leads, which eventually led to the development of protease inhibitors. Can you give me the example of a protease inhibitor, guys? A class of drug that become integral in HIV treatment. HIV protease enzyme inhibitors. So first they targeted the HIV protease enzyme. They found multiple hits, and then those became the potential lead, and that lead became the drug, right? So can you give me the example of HIV protease inhibitors? Yes, guys. Anyone? Cyclovir, flecainide. Any other drug? Oseltamivir. Okay. Andinavir, sequinavir. Yes. Okay, so hit identification is done. Now, the second step is done. Now, the third thing is target validation. Now, we have validated the hit. Now, target validation. Okay, target validation is also the similar process and the parallel process. Okay, target validation is, I mean, it is a similar process. Like in this, we confirm the identified target as a significant target. Identified target as a significant target. I just gave you the example of COX inhibitors and P2Y12 inhibitor, right? In the disease, and that is modulating this target. So what are the methods for target validation? We check with the genetic manipulations. We check with the genetic manipulations, like which is most expressing or overexpressing or underexpressing, right? We check through the genetic manipulations, like we use the techniques like CRISPR, RNA interference, RNAi, or gene knockouts can be used to disable or modify targets in the cell or in animal models to see if the disease symptoms change. So how we are identifying the targets and how we are validating the targets. Now we identified the targets in the first step. In the stage now, we are validating if this is the correct target. If we are not, just faked or fooled by something in the body, we should not do that. We should check it again and again. So we check it in the animal models, we check it in the cell line, we check it everywhere just to check if this target is actually changing or showing the symptoms when we are doing it, when we are manipulating that. Okay, so genetic manipulation can be done, pharmacological inhibition can be done. So we are using the small molecules or antibodies to inhibit or activate the target in experimental models to observe the effect on disease progression. Again, in the animal models or cell lines, we check that pharmacological inhibition. If this target is actually, helping, or is this target is actual target, what should be targeted actually to, interfere in the disease progression or to suppress the disease or to treat the disease by maybe small molecules or antibodies or anything. Right. And then we can check the biomarker studies, like, if there are any changes in the biomarkers, which is directly correlated with the target. Okay, so in example, if you guys can read it, if you guys can see it in the, as I was giving in the last slide also, chronic myeloid leukemia. This is a cancer. Scientists validated BCR-ABL. It is a mutated tyrosine kinase, tyrosine kinase enzyme, mutated tyrosine kinase enzyme as a target. Experiments showed that inhibiting BCR-ABL in cell line and animal models of CML could halt cancer cell proliferation. It can stop the cancer cell proliferation, and this validated led to development of imatinib or amoxicillin, a highly successful drug targeting BCR-ABL. Right. So in CML, the chronic myeloid leukemia, scientists validated BCR-ABL, which can be treated with imatinib. So BCR-ABL was target, which was validated as the tyrosine kinase inhibitor, and tyrosine kinase inhibitor is imatinib, the drug. You understand? So that's how we validate the target in the animal models or in the cell lines, okay, which are mimicking the human beings, okay? Target validation, right? So look at this, guys. I wanted to show you this. There are multiple things what you can try in your libraries. This is the open slide for all of you. You can take a screenshot. There are druggable and undruggable human proteins. Okay, there are total of human proteome. There are 17,294 proteomes, which are actually, we have seen till date usually, like in all the drug research, 17,294 proteins, and disease-related proteins are 4,444 are the disease-related proteins. Okay, in that, druggable are only 854. The druggable proteins are, or druggable proteins are only 854. Okay, in that proteins, these are all everything is almost protein. So there are 323 enzymes, 323 transporters, 111 receptors, and 110 GPCR. Okay, 110 GPCR. So these are druggable targets. These are druggable targets. These are druggable targets, and these are difficult to drug target. These are very difficult. We cannot target, like where there is a lack of hydrophobic pockets, where there is lack of hydrophobic pockets. There are functions via protein to protein interaction. We usually don't target protein to protein interactions. And then active sites are highly conserved among family members. Highly conserved means there are no space for other things to work or attach on the proteins. So where there is a highly competitive area for the receptors, where there is highly competitive area for the receptors on the drug targets, that cannot be the usual druggable targets. We just leave them because there is no space for drug to work, right? And then where there is a lack of deep grooves and active pockets where the drug can actually attach, where the hit or lead can directly attach and affect. If there are lesser of these things, these are difficult to drug target. And out of 17,000, only less than 1,000 are druggable targets. Okay, this is the hint for all of you. This is the hint for all of you. Like if you want to do the research, if you're doing the research in that, definitely you will be, searching for more. Okay, so that's the target. Now, importance of target validation. Importance of target validation is very important. It's identifying and validating targets accurately is essential because it increases the chances of developing an effective drug. If we are having the perfect match of target and lead, what we will get? A drug. We will get a drug, right? So that's why validation of the target or validation of the hit is very, very important. So we get the most relevant item out of all these. If a target is not properly validated, a drug might fail in the later stages. If the target is not validated multiple times, the drug might fail in the later stages. So by ensuring that a target is both relevant to the disease and druggable. Look at these two terms here. Both, one is relevant to the disease and druggable. Scientists can proceed with the more confidence towards screening the compounds and drug development. If the target is validated, we can go further, to the next stages. So now this is the wonderful thing. I kept this today only. Okay, druggable and undruggable human proteomes. This is not the target actually. This is not the title actually. The correct title is use of AI ML in drug targeting. Okay, or in early preclinical stages. Okay, the title of this is AI, artificial intelligence and machine learning approaches in drug targeting and drug discovery in early preclinical, very specifically early preclinical stages. Okay, so there are two types of AI ML approaches. One is supervised, one is freehand. One is supervised, one is freehand. We just leave the freehand based on the clustering, based on the clustering of the data, based on the clustering of the data. As we were talking about virtual screening, I'll not go to the deeper of that. I even don't know much, but I mean, the basic things I know. So it is unsupervised means freehand. This is the freehand where we work on the clustering, and this clustering works on the molecular designing by finding the k-mean, hierarchical clustering. Right? These check which targets are most relevant with the hits or most relevant hits with the target. Again, in the virtual system or in the virtual environment, right? This is unsupervised freehand AI ML approaches to discover the drug. Okay, and there is something called hidden Markov. There is something called Markov model, hidden Markov model, which again works in the selection of features or selection of properties, property match. This is called property match, property of a chemical entity and the target, which matches by the hidden Markov model with the virtual compounds. Okay, this is the AI and ML approaches, which is unsupervised or freehand. One is supervised. In the supervised model, we usually go for the regression model or classifications with NLP. Okay, NLP. Can someone tell me what is NLP? Not natural linguistic program. This is not natural linguistic program. NLP, and then SVM, drug targets association, we find with an NLP. Okay, we find the drug targets association with NLP, and we look for the nearest neighbor also. We run the Bayesian networks also to target the, I mean, to identify the target or to find the target druggability. Okay, and in the regression model, linear regression or decision tree or by random forest plots, we find if the disease drug response are common or if the disease drug response are meeting the criteria or disease target druggability is meeting the criteria if they are responding well to each other, right? So we check in the uncontrolled and controlled methods with the help of artificial intelligence and machine learning in the drug discovery. Okay. So then lead optimization. So lead is the key stage in drug discovery that follows the hit identification and lead identification. It involves refining the promising compounds from 10,000 to 1,000 or 100 to 1,000. Okay, this step is essential for enhancing a compound's drug-like characteristics. Compounds drug-like characteristics before moving it into preclinical and clinical settings. Okay, so first thing in this, there are multiple steps. One is first enhance the potency and efficacy. In this, actually leads are optimized. In this, I mean, till now there were hundreds of most relevant chemical entities found. Okay, and now we are refining them to become a drug which can be used in the preclinical studies. Okay, so first enhancing the potency and efficacy, that definitely is done in the laboratories, chemistry laboratories, analytical laboratories, pharmacology laboratories, and these can be done in all the pharmaceutics laboratories. So this is the laboratory work. This is not even in the animal models or cell line. This is not in vivo. This is completely in the laboratories. So here we have some drug components and we are optimizing that to what? To check the best effective with the target. If it is able to bind with the target and produce a strong biological effect on the target. So chemists usually make small structural changes to lead compound to improve its affinity for the target, which typically increases the potency and reduces the dose needed for effectiveness. Okay, so now we have started working on the actual things like increasing the potency and reducing the dose. Okay. Second thing, again, as I told you, selectivity, improving the selectivity. So selectivity refers to a compound's ability to interact specifically with the intended target without affecting any other similar molecules or pathways. Right. So selectivity is very important. So here we just minimize the off-target effects. We in this improving the selectivity in the labs only, we minimize the off target effects. Okay, reducing the likelihood of side effects. Here we are resolving the issue of side effects to the most. Okay, so it cannot affect, like, okay, I'm going to my house, but I should not go to another house which looks similar to my house, right? If I am going from place A to place B to some house, I should not go to other houses which are looking like similar house, right? So that's what we do is we do the lead optimization here. We minimize the off target effects or off target directions here in this to reduce the side effects. Okay, by testing the compound's effect on related proteins or enzymes, scientists can design the modifications that minimize the interaction with unintended targets. Then optimizing the pharmacokinetics. This only lead optimization also goes with absorption, distribution, metabolism. So here we ensuring the compound can be absorbed effectively in the body, typically through the oral or intravenous administration, right? Distribution, making sure that the compound can reach and concentrate at the target site. These are distribution, absorption, metabolism, excretion are the special science. So I'm not going to that. But here in the lead optimization, we are optimizing the pharmacokinetics. Metabolism, modifying the compound to ensure its suitable metabolic profile, balancing stability and metabolism. Excretion, ensuring the compound is cleared from the body in a predictable way to prevent the toxicity. Right? So this is all is lead optimization. Then reducing the toxicity in cell cultures, animal models, and we are modifying the lead chemically, and which are we are modifying the lead chemically by removing or replacing the toxic parts of the molecule, while maintaining the activity. Reducing the toxicity in early stages is essential for preventing harmful effects later in the development, especially during the clinical trials. So reducing the toxicity, then formulation and druggability check. So lead compounds are optimized to ensure that they have good solubility, stability, and compatibility. So we can go ahead to make a drug, to make a test drug. Scientists aim to create compounds that are easy to manufacture, stable under various conditions, and suitable for intended route of administration, oral or injectable, anything. Lead optimization, okay? And then now preclinical studies. So preclinical studies, I think all know, preclinical studies are in the in vitro or in vivo. So in vitro is nothing but in laboratories, but again, used on the cell lines or not directly in the, these are not in the live animals or human beings. Not definitely preclinical studies are in animals, but these are not in the live animals. Okay, these are on the tissues or cell lines, in vitro and in vivo, to evaluate its safety and efficacy and pharmacokinetics. Again, because we have to give it to the human beings. So till now, what has happened is we found a hit, we made it lead, which is most suitable in laboratory. Guys, focus in laboratory. Just 10 more minutes. In laboratory, we made the best drug in laboratory without using any similar components to the human beings. Now, once we have lead and target, target is different thing. I'm just talking about the drug. So hit to lead, and lead, we just modified, we just did all the alterations in laboratory and make the best lead here, best lead. And now this is the time to take this best lead to the best test in the similar parts or similar cell lines or similar animals to get the best efficacy and potency. That is called preclinical studies, right? So preclinical studies, there are goals. One is assessing the safety. So we determine the compound safety by finding if there is any toxic effect on the various doses. So here dosing also is doing. Okay, doses also can be done here. Dosing not only here, but in the clinical also. I'll tell you that tomorrow. There is a big phases, you might have not even heard of. I'll take you through the very deeper phases of that. Okay, so any toxic effects we remove, evaluate the efficacy by compounds effect, against the cell cultures or animal models, against the disease, we check the efficacy. Then we again, study the pharmacokinetics and pharmacodynamics to again check if it is the best interacting with the receptor or best metabolizing or absorbed or distributed in the body. Okay, then type of preclinical studies, one is in vitro, which can be done using the cells or tissues, or to assess the biological activities binding to the targets, potential toxicity in various cell types, right? And in vivo, like directly in animals, right? Different animal models like rats or mice, sometimes dogs or primates, right? So in animal studies, we go for acute toxicity, chronic toxicity, efficacy, dose ranging studies, pharmacokinetics, pharmacodynamics. We check all these in the preclinical studies. That is the pharmacology part. I'll not go to that. Okay. Then this is all for today, right? Toxicity, when we are checking the toxicity, we look for the genetic toxicity, we look for the cancerous toxicity like carcinogenicity, we look for the toxicity on reproductive and developmental uh, science, if it is affecting our if it is affecting our reproductive power, right? So we check these three major toxicities when we are working in the preclinical studies. We look for the genotoxicity, carcinogenicity, and reproductive and developmental toxicity, right? So example of preclinical testing: in vitro, testing the drug on various cancer cell lines to see if it kills or inhibit the growth of tumor cells. In vivo, we give the drug to the mice with induced tumors to evaluate if it is affecting the tumor growth and affecting or reducing the spread. And for the toxicology, we study whether the drug causes any damage to non-cancerous cells or non-cancerous tissues in that. Now I'll give you a best example, guys. I will summarize the whole presentation. I hope you're liking it. I will just summarize the whole presentation with an example, full example. Okay, so the example of lead optimization here is in case of atorvastatin. You know, so atorvastatin is a cholesterol lowering drug. Lead optimization involved enhancing its binding affinity to HMG-CoA reductase, that is enzyme responsible for cholesterol synthesis in the liver. So atorvastatin, like the statins, work usually on HMG co-reductase, right? So it inhibits that. Scientists modified the chemical structure to improve its potency, resulting in the high effective compound that requires lower doses, and they came up with rosuvastatin. First, they came up with atorvastatin, then they again reduced the things, they again modified it, and they came up with rosuvastatin, right? So there are a lot of examples. Now, this is the example summary of the entire thing. That was the another example. This is the example what I was talking about. See, full drug discovery you can understand by this example. Okay, you can understand this all by this example. Example of drug discovery and preclinical research can be seen in the development of imatinib. Imatinib is the drug, anti-cancer drug, which is used in chronic myeloid leukemia, right? So see, first how the target identification and validation was done. Scientists identified a specific protein, BCR-ABL, which is a mutated tyrosine kinase enzyme that causes uncontrolled cell division in CML, like chronic myeloid leukemia. This enzyme results from a chromosomal abnormality known as Philadelphia chromosome found in CML patient. So in CML patient, they found the Philadelphia chromosome, and that helps in chromosomal abnormality, and that helps overexpressing BCR-ABL, that is a tyrosine kinase mutation. Okay, mutation enzyme, right? So this Philadelphia chromosome helps tyrosine kinase enzyme that actually overexpressed BCR-ABL, and this is the best therapeutic target then. So we can block this to stop or to slow down the cancer progression. BCR-ABL is the target identified, and it was validated because it is helping, BCR-ABL is helping the, what we say, progression of the cancer, and it is being done by Philadelphia chromosome, and it can be stopped by tyrosine kinase enzyme inhibitors. Okay, so next, second thing was hit identification. First we had target is BCR-ABL. Now if we go to the hit identification, researchers screened various compounds to find hits that could inhibit BCR-ABL protein. After identifying promising compounds, they optimized these hits to improve potency, safety, and bioavailability. The compound imatinib emerged as the lead candidate due to its ability to specifically inhibit BCR-ABL while sparing other kinases and minimizing the side effects. So imatinib was the best hit because it was just, it has the ability to inhibit the BCR-ABL, but not affecting the other kinases. Okay. Then in the preclinical studies, in vitro studies, imatinib was tested on cancer cell lines with the Philadelphia chromosome. These studies showed that the imatinib could effectively inhibit the BCR-ABL and prevent the growth of CML cells. In vivo studies, animal models of CML were used to assess the drug's efficacy and safety in living organisms. Imatinib was shown to reduce the tumor growth with manageable side effects. Pharmacokinetics and toxicological studies. Additional studies were conducted to understand how the drug is absorbed, distributed, metabolized, and excreted. These studies ensured that the drug had a favorable safety profile for human clinical trials. Okay, and with that, this was came into the existence as a drug for CML. Okay, guys. So with that, I'll end the presentation. I'll end today's topic. So we are done for the today's topic, and I will also come up with this. I'll also take this course. This also I have designed for all of you. This is also a 6 days course. This is also a 6 days course. Public health research and epidemiology, fundamentals and applications. I'm launching first time for you only. Okay, it is not launched in any groups or anywhere. So it will also has the 6 days. First, we'll be talking about public health and epidemiology, study designs in epidemiology, data collection, interpretation, outbreaks, investigation, public health interventions, global health and epidemiological challenges, practical applications, and that. Okay, so this course is also there for you. This will start on 12th December and end on 18th December. There is still time. So again, the same timing, 7 p.m. to 8:15 p.m., Monday to Saturday, 6 days. 12 to 18 December. If you are interested, the same price 199. I don't want to keep the prices high for anyone because of this. So if you guys are interested, you can also register to that. Okay, epidemiological studies, right? Okay, guys. So now I am here for your questions. Please suit up the questions. Please suit up the questions. If you have any questions, please come up with the questions, guys. I'm looking forward for the questions. And if you are interested, you can even register now for the epidemiological. I just shared the link here, and I'm sharing the link for your today's class also. Attendance. Wait, guys. I'm sharing the attendance form also, and then we can discuss over the topics and the questions you have. Okay. What's the first day? This is, I believe, today's only. Yes. This is today's. Okay. This is today's form. So you can just fill that. Guys, fill it and please come up with the questions. I'm here to ask, to answer the questions. I'm not asking now. I'm done with asking. Now you have to tell me. You have to ask me the questions. Yes, guys, fast. You have to fill this form in 5 minutes. You don't have more than 5 minutes or 3 minutes. Not 5 minutes, 3 minutes. You have only 3 minutes to fill this form, and you can come up with the questions now, guys. Please. Yes, everyone. Questions, please. Now let me open your mics first. Oh, I'm so sorry. Guys, can you please open their mics? Someone from my team, can you open their mics? Okay. Your mics are, I think, open. You can speak, guys, if you want. You have questions, please speak or write in the chat box. And fill the feedback form. That is the attendance form, actually. Fill the attendance form. Yes, guys. Any questions or clear? Everything is clear? Come on, there are 150 people. Is everything clear for you? Or any questions, guys? Please ask if you have any questions. I hope you like the class. Yes, guys. Please ask me if you have any questions. Thank you. We have to cover a lot in 6 days, guys. So I, request everyone to go ahead and read more. If you are interested in this, please go ahead and read more about it because this is very big, vast topics we are talking about. I still feel that we are talking basics only. Okay. So if you want to know more, go and read the examples. The best part to understand this concept is examples. As I'm learning the examples, I'm giving the examples, I know the examples. So you also must go through the drug profiles, how they have been come, to the place. PDF file, no, 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 guys. That's the problem. I will not share the slides. I will share the recordings. You will have the recordings. You can take the screenshots. Nowadays, there are the AI models. You can take out the content from the slides. Okay. What will be the followed for drug used in emergency situations like corona vaccines? Wonderful question, Sitaraman. I will take this example in the next class. Okay, next to next class. In 2 days, I'll take this example in the regulatory part. Done. Okay, I have that in mind, how the corona thing has come and how they came up with the vaccines, what they did in the fast track. We will discuss that. Okay, we'll discuss that as an example. Yes, any other suggestions or questions, guys? Yes, anyone else? No questions, really? You understood everything? Okay, fine. Was there? If you understood everything, I don't mind. I thought you will have a lot of questions. How I was teaching, I thought I'll give you a lot of curiosity and you will ask more questions. I really thought that I'll come up with a lot of curiosity and you will ask me a lot of other questions. But you're not asking. That's surprising. Okay. Anyways, guys, have a good night. And I took a really long class. It's almost 120, not 120, almost 105 minutes we have spent together, 100 minutes, more than 100 minutes. So, okay. Bye bye, guys. Take care. Good night. Thank you.